Okay. Hello, everybody, and thank you very much for the opportunity to speak here in this session. Today I'm talking about how we utilize the quartz crystal microbalance in order to study tribofilm formation in cobalt chrome molybdenum, on cobalt chrome molybdenum alloy. Our study was funded by, or our ongoing study is funded by NIH and NSF. Many people already talked in this session about cobalt chrome molybdenum alloy, and as you know, it's extensively used for orthopedic prostheses, especially for articulating surfaces, but also in modular junctions. And you also all know, especially when you visited Dr. Halab's talk a couple of days ago, that there's enormous concern due to implant failure caused by adverse tissue reaction as a result of excessive wear debris generation or ion release. However, there's also earlier findings that pointed to the occurrence of a carbon-rich tribofilm. You can see it here in the corner, that thinner gray layer. This carbon-rich tribofilm adheres strongly to the surface and was especially observed in first and second generation well-functioning metal-on-metal bearings. But there's still a lot of questions. For example, how does this film form? How does it really influence the friction and wear behavior? Is it a beneficial layer or not? One of the problems we have is that it's difficult to generate a uniform tribofilm in order to, have some in vitro investigation of its fundamental properties, tribologically, mechanically, etc. So what do we know so far? Let me quick summarize. We know that this layer is presumably generated from protein in our solution or in the synovial fluid, mainly due to strain. Further, we know that it contains metal ions, cobalt, chrome, molybdenum, and also nanoparticles, like cobalt chrome particles. In an earlier study, we have shown that this film not only lies on the surface, it even interacts with the nanocrystalline metal surface kind of through mechanical mixing and gets incorporated in the surface. Another study has shown that there's even a partially graphitic found, which you can see here by these eel scans shown by this pi peak in the eels plot. I would like to point out at this point the study that has recently been published just online a week ago in Langmuir by my colleague Elizabeth Martin, and she has data that makes us strongly believe that there's also an electrochemical influence that that enhances the formation of a tribofilm, and she talks about the significant role of molybdenum ions to this process. Since these results are rather new and they're kind of important in order to understand our recent findings, I kind of want to talk about it a little bit. So for those of you who are not familiar with the quartz crystal microbalance, I don't know how many there may be. So here you see such a quartz crystal. It has two electrodes on the front and the back side, and placed in a QCM holder, we can apply a voltage and a frequency sweep, and what you get as a result, you get a conductance peak at the resonance frequency, and each harmonic, art harmonic of this frequency. And why is this important? Well, this frequency, this conductance peak is very sensitive to mass changes, uniform mass changes on the surface. What it means is a negative frequency shift means a mass increase, a positive shift is mass loss. And we can quantify these mass changes by the Sorby equation. So we're not interested in the corrosion properties of our crystal. Therefore, we first have to apply a cobalt chrome molybdenum coating to our crystal. Here's a cross-section in the FIB. In our case, we have a stable coating at between 200 and 300 nanometers. Other groups I have seen, they achieved thicker coatings, which are stable. We verified that this coating is relevant for our application, so it's a nanocrystalline coating, hexagonally close packed, exactly like the subsurface structure in metal hips during steady state. So the next step was to integrate this quartz crystal microbalance to a 3-electrode corrosion setup. So we use the surface as our working electrode. Here's the quartz crystal holder, and here's the reference electrode and counter electrode. Here's the resulting plot you get from such an experiment. This experiment was in a buffered solution with no protein. We also did the same experiments in Ringer solution with similar results. Let me quick explain. Your red curve is your typical potentiodynamic curve. You have here the potential on the x-axis, the current density on the y-axis, and the blue curve is now your mass that you can measure in situ during the test. And you can see during the passive regime, the mass at this scale is relatively stable. Once you hit the transpassive region and you have a current increase, you also have a monotonic decrease in your mass. So that's your corrosion mass loss. The surprising finding was that in the transpassive region, though, that when we do the same test in bovine calf serum that we use for wear tests with 30 gram per liter protein, we had a significant mass increase. And taking the sample to the SEM and looking at the surface, we find a carbon-rich film on the surface, a relatively strongly adhering carbon film. And we also performed XPS and found similar results than we know from our tribofilms that are formed in vivo. And that led us to the conclusion that there might be an influence of electrochemistry on tribofilm formation by ion protein binding. So for the second part of the talk, I talk about the two goals that we had. First of all, we wanted to verify similarities between these tribofilms and those films that we generate electrochemically. Further, we investigated the wear and friction properties. We wanted to investigate these properties of cobalt-chrome lipid surfaces with and without such a film. Therefore, we had to establish a new wear test rig in which we also wanted to utilize QCM for mass monitoring. Okay, for the first goal. Here you see again in the FIP a SEM picture of our carbonaceous film. We utilized the tungsten needle in order to scratch off a little sample from our film, placed it on a TEM holder. That's our little sample here. And then looking at it here, you see that the film sample is a higher magnification of this area, and there we were able to apply yields. And when we do a global scan of our sample, you can see we have a clear carbon peak and a smaller nitrogen peak, indicating indeed the proteinaceous nature of this film. When we look more localized at several spots, you also can find, even though it's small peaks, there's still significant chromium and cobalt. And also at several spots we found this pi peak indicating graphitic nature. We also had performed Raman, which further confirmed our assumption that this electrochemically generated film can be considered a tribofilm-like material. So the good thing about that, now we can very controlled, either on the quartz crystal but also on bulk samples, generate a film which is similar in chemistry and structure to tribofilms. And we can test it in a wear and friction test. So this is kind of our setup. We more or less misused the rheometer as a pin disc type of tribometer. Our disc was a sputter-coated quartz crystal embedded in the holder. We had as a counterbody here three pins with spherical tips. They applied the normal load on the vertical axis. We applied two motions, like first of all a rotational motion and a reciprocating motion. And this is because the quartz crystal microbalancer, the quartz crystal is most sensitive, first of all in the middle, and it works the best when you distribute the contact over this entire sensitive area and not only localized in one spot. So one thing I have to mention, though, we don't have real in vitro mass monitoring like in the corrosion test, because every time at certain points of our experiment, we have to lift off these pins out of contact, measure our QCM, with our QCM the mass changes, and then restart the experiment, which goes rather quick, and it's better to take it out and take weight measurements or profilometric measurements and so on. So in our pilot test, we had three different testing conditions. We had one in distilled water, we tested in bovine serum, and the third test was in bovine serum, and we pre-applied an electrochemically a tribofilm-like layer. Let me first show this plot which shows you the friction, and you see for the first test in water you had like relatively high friction and extremely high scattering of your friction signal. And bovine serum, you see the green curve already has significantly lower friction, but also still scattering. And the red curve shows the film as a sample with an initially applied film. And you can see that initially the friction coefficient was lower. I want to look at test 2 and 3 a little bit closer and look into our mass monitoring. And you see in test 2 in bovine serum, the mass was initially relatively stable, but then eventually decreased relatively fast as the friction scatter also increased. And the reason is a problem in the system, in our setup, that due to the high surface pressure, eventually we have a fatigue of our crystallines. Bigger particles break out and penetrate our coating, as you can see here. So we have an increased three-body wear, which kind of is the reason for this fast mass loss. However, we found no tribofilm formation, so no film formed as in this test. In the second test with the film, initially applied film, you see that the friction, the lower friction, so you have a kind of a startup friction that's relatively high, but then you have lower friction as this carbonaceous film gets rubbed out of your contact. Then it seems to have a saddle point and eventually undergoes the same three-body wear as in the first test. Again, this tribofilm only had an initial effect but didn't retain itself during this test. Here's just another way to look at that data. Test 2 and test 3 in the beginning and in the end of the test. You see test 2 basically had the same friction over the whole time in average, just the scatter changed. But the friction force was, for the test with carbonaceous there, significantly lower in the beginning of the test with less scattering. And in the end, it approached kind of the same values as the second test. And you can also see here that when you look on the surface, that as long as you still have film, your friction is lower. What are our conclusions? First of all, the tribofilm formed in vivo is almost identical to the electrochemically generated film. We also successfully utilized the QCM as a mass detector during wear test. The tribofilm appears to be a solid lubricant. And concerning our setup, the effect of the tribofilm was not durable and it could not be formed during the wear test. There's two reasons. Basically, our Pinot disc setup is not suitable. We have too high contact pressures and non-conforming surfaces, so that lubricant is kind of rubbed out of contact. We assume that conforming contact conditions at those metal-metal hips seem to provide better conditions for tribofilm formation. In summary, tribofilm formation appears to be dependent on iron-protein interactions, as they may occur during tribocorrosive processes in metal hips. Once formed, it will reduce friction. Our ongoing work, we are modifying our test rig. We want to introduce a ring-on-disc contact that has several advantages. It will reduce our surface pressure, which is important. We are like tenfold of this right now. Then we hopefully, by doing this, will also reduce fatigue of the crystal and reduce three-body wear. And we expect that this will also help to generate a film, since you have these conforming conditions. We are introducing also a tribocorrosion setup to this whole system, and we are investigating the influence of single protein species on that film formation electrochemically. And another big question is, okay, the tribofilm seems to be good for our coefficient of friction, but does it have biological consequences, since you generate a kind of metallo-organic complex on the surface? I'd like to thank our funding sources, the Schall Group at Northwestern, where most of the work was conducted, our collaborator Dr. Fischer in Germany, and Orwag for providing the alloy for our experiments. Thank you very much.